So over the past few months, my partner Rachel and I were able to use a multi-method approach to hypothesizing a function for the gene Bradi1g64770 in Brachypodium distachyon. And we were able to use a variety of experimental methods ranging from in lab experiments to online database research. And this was for a variety of reasons. It gave us a good background on the different research techniques that you can use to develop hypotheses for a gene, but also because we needed to come up with a good body of evidence surrounding what we believed the function of our gene to be. So the model that we worked with was right here, pictured on the right. That's Brachypodium distachyon. So we planted at the beginning of the semester. We started with a group of seeds from a stock of induced mutations. And these mutations were believed to have an effect on cell wall growth. So throughout the semester, we used those plants to experiment to visualize the effects of that mutation. And there's a variety of reasons why we use Brachypodium. First of all, that's what we were provided. But Brachypodium, it boasts short germination times. It grows quickly in the lab. It's fairly easy to work with. All of our plants are able to germinate successfully and grow. There's few ethical issues and regulations surrounding research with plants, so it was easier for us to, be able to work with this organism. And then it was simple to store, and there wasn't a lot of training involved in working with this organism. So there wasn't any kind of special assistance we needed. When we need to harvest a sample, you could just go in and take a clipping with scissors, and that was really all you needed to do. So the first model we constructed the ab initio and the homology-based model. And basically, we compared the sequence to we ran through F-Ganesh, and we came up with basically the contiguous sequences in the exons that we saw in our sequence. And up here, we see all those exons that we have, and then we align that on BLAST. And then so on BLAST, we compared our sequence to the data that was available, and we saw that there was a lot of good alignment. So the red represented those good alignment scores. And there was some good taxonomy data relating similar alignments to other species. There was not a lot of similar alignments in Brachypodium on BLAST, but there was definitely a lot of similar proteins in other organisms. So mostly tobacco plants, rice, and Arabidopsis. And that's what led us to our library search. So the first paper we were able to look at was a review of our gene, which BLAST returned. It was very similar to the KCH domain in plants. And the KCH domain, basically, it has to do with microtubule binding and microtubule growth. So this is from a review paper about this domain, where these are two different views of a cell here. And these red, that represents our microtubules, and then the black here, that's what they use for the KCH domain. And basically the KCH domain is what's responsible for the growth, the extension, and in these microtubules. And then the second main function of our gene that we found through the literature was the nuclear localization. So nuclear localization in these cells. So the first experimental paper that we looked at was right here. So these two graphs represent, we can look at mean cell length and mean cell width in two different lines here. One, a wild type, and a KCH overexpressed version of a similar plant. So comparing the two, we see that the cell length is affected by the overexpression of KCH, while it's not the same for the cell width. And that's supported again on the right here with micrograph images of these cells, where we see the differences in the wild type cell and then the KCH overexpress. And then for the second function of our gene that I just talked about, it's the nuclear localization. So the x-axis on this graph represents the relative position, where 0.5 represents a nucleus localized to the center of the cell, and anything less is further from the center. So basically in the wild type, we see that there is strong localization, the nucleus is most often found in the center of the cell, and then when we overexpress our KCH domain, we see different results.
The phylogenetic analysis of our gene was useful to narrow down most likely function based on similarity of Arabidopsis, Orza, and Solanum plants. Respectively, these grass-like rice and tomato plants are very closely related to Brachypodium with common ancestors between many genes. In this tree, A. trichopoda is used as an outgroup. Highlighted in yellow on the tree is our gene of interest. The homologous rice gene encodes kinesin 14, as does the orthologous tomato gene with similar common ancestors to the Brachypodium gene. Scientists know much about the Arabidopsis genome, therefore these homologs were most important in narrowing down the function of the Brachypodium gene. The AT5G27000 encodes kinesin 4, whereas AT1G64770 encodes P-loop nucleoside triphosphate hydrolases superfamily in the CH domain. This supports our hypothesis that our gene contains a kinesin 4-linked CH domain. We used electronic fluorescent pictographs to determine parts of the plant and stages of growth with most basal expression of our gene. This atlas is constructed to map all RNA data pertaining to our gene anatomically and developmentally. The middle-length grain, young seedling, and internodes near the bottom of the stem showed most gene expression. The scale in the upper right corner indicates abundance of gene expression, with bright red being most abundant. You can see the most constitutive expression, like we said, in the middle length and young seedlings, and this is seen most near the first internode at the bottom of the stem, according to the pictographs. The stem grows from the first internode, and the first node in the internode of the plant at the first 2 centimeters of the peduncle, and this indicates our gene plays a role in stem growth. This is confirmed by the fact that there is moderate to high expression in younger versions of the grain, and that an increase in expression is seen again when the grain transitions to a growing seedling. We analyzed experiments from the Hazen lab to determine temperature and light cycles' effect on our gene. It was determined that our gene is affected by thermocycles, which is confirmed to be statistically significant with the plot on the right. However, we did not find this information useful in determining gene function further. Pictured on the left is a t-test performed comparing gene expression in roots, stems, and leaves of the plant. Our conclusion is based off of the p-values calculated from the t-test with the threshold of 0.0167 according to the Bonferroni correction. All p-values calculated between tissue types were much less than this number, allowing us to confirm the difference in gene expression is not due to chance, indicating our gene is expressed much more in the stem than the root and leaf. This is consistent with our previous idea that our gene is expressed most at the bottom of the stem where a plant grows from. Moderate expression in roots also supports this idea. Once our gene has been amplified and purified from mutants and wild-type plants, samples were sent for Sanger sequencing to confirm the presence of the NAN152 mutation in our models. From gene modeling, we were able to determine the deleterious gene causing physical mutant effects. Previously stated, the NAN152 mutation is a splice donor site substitution. Where there would normally be a G nucleotide marking an intron cut site, an A is substituted. This ruins the majority of proper gene function, and it introduces a stop site codon, hindering the gene from encoding the rest of its needed proteins. As this is a homozygous mutation, we expected all mutants to show homozygous sequence results. Unfortunately, our gene sequencing was inconclusive due to cross-contamination or possibly due to low DNA concentration after purification. The Tuesday-Thursday section had inconclusive results as well when sequencing the same gene as us, and there was no data from previous years to confirm the presence of our expected mutation. Thanks to the Hazen lab, cross-sections of wild-type versus mutant plants with our NA-N15 mutation in our gene were available. The cross-sections shown are from the stem tissue of wild-type and mutant B. distachyon plants stained with fluoroglucinol HCl. After determining likely function in different cells of our plants, we chose to compare epidermis cells, figures A and B, and interfascicular fibers, also known as IF cells, shown in figures C and D. The IF cells of the mutant appeared to have thicker cell walls and darker staining compared to the wild type. The epidermis cells of the mutant appeared to have thicker cell walls as well, but the same staining compared to the wild type. After qualitative observations, we used a statistical analysis to determine whether differences in the cell wall thickness were due to chance or not. We performed t-tests to discover differences observed in epidermis cells were due to chance with a very high p-value. However, differences observed in IF cells were statistically significant with a p-value of 0.01958. Unfortunately, our small sample size affects proper statistical analysis, but the distance of p-values from the 0.05 threshold leads us to believe that our gene plays a function in IF cell growth, but not epidermis cell wall growth. Between gene expression statistics, known literature, and phylogenetic analyses, we can determine the likely function of our gene, Bradi1g64770. Once we determined a homology-based model for our gene, we were able to determine known mutations in literature based off of similar gene sequences. This literature led us to decide our gene most likely contains the KCH domain, kinesin with calponin homology. This protein functions in cellular division and growth, gathered from multiple articles. We hypothesized it scaffolds proteins and cellular components to prepare for cellular division, as well as signal growth during interphase for cell division during mitosis. To narrow down specific function, we created a phylogeny for our gene and analyzed Arabidopsis homologs, confirming our hypothesis. The Arabidopsis homologs encoded kinesin and P-loop nucleoside triphosphate hydrolases superfamily protein with calponin homology, which both function together in microtubule motor activity and ATP binding. We further confirmed this using electronic fluorescent pictographs indicating our gene is most expressed in the first node and internode of the plant, where growth stems from. IF cells are key in stem growth and from the internodes, which is why we see differences in IF cell walls between mutant and wild-type plants. In conclusion, our gene functions in cell wall growth of IF stem cells. Specifically, we believe our gene assembles kinesin and microtubule proteins with ATP to drive motor activity in the cell during stem growth. Overall, this affects new stem growth of the plant, causing mutants to be smaller, which we visually saw with our plants. In the future with this research, we can further add to the known Brachypodium model genome. Model organisms like Brachypodium are important for initial clinical and genetic research in other organisms. Discovering more about the KCH domain encoded by Bradi1g64770 could lead us to understand more about microtubule motor activity contributing to growth in other organisms, which could eventually even have clinical implications. We would like to thank Professor Hazen as our instructor and director of our research, and for allowing us to use Hazen Lab to complete our project. We would also like to thank Professor Razaford for additional guidance in this project and help with statistical analyses. We want to thank Katie, our previous lab TA, for our help, and Katie, the lab coordinator, for hers as well. Lastly, thank you to UMass College of Natural Sciences for allowing us to complete this project with excellent tools and staff. And of course, thank you to our classmates' help and support during and throughout the semester.